We're solid on the dominant recessive codominant story, or incomplete dominant story, and let's just look at this idea a little bit. So I made up this addition to your book, and that is that here's an allele that we've learned about, then allele that we've learned about, and then we'll say here's another allele for that same gene. And so I just set this up to show you that we have to deal with these alleles as pairwise combinations. So in this case, allele 2, we learned, is recessive to allele 1. So we went through this and we learned about this, that purple is dominant to white. White can be a carrier, can be hidden, and that's the case when we do the crosses. And we had to do the breeding experiment to show that, to learn that over time. And that this allele 2 is recessive in this pair of alleles, but in another pair of alleles of the same gene, this allele may be dominant. So it's just a little bit challenging, but that that allele 2 is recessive to allele 1, but dominant to allele 3. And this, I just made this up. I just made this combination of alleles, and this is a possibility that you do these crosses, and we do the breeding experiment, and we find that in this case, we do the breeding experiment and find these ratios that allele, the white allele, is allele to this lethal allele. And that just brings up the concept of that this allele may be recessive in certain situations, it may be dominant in certain situations, it may be incomplete dominant in certain situations, and what other situations the situations are in pairing with other alleles from that gene. And then also just this idea that we talked about this with hemoglobin, that this could be pleiotropic, where in this case, this gene affects pigment, but there's some alleles that are that could lead to death, and it's just because genes often play the same functions in multiple different settings, and so this may be a gene that codes for an enzyme that's important to make the pigment, but maybe the enzyme in another cell, another type, that actual chemical reaction on the pathway toward the pigment may be on a pathway to another essential product. And if you don't make that product, you're dead, like that. So that's just bringing out that concept of multiple alleles and the combinations of them. Now, then, here's the next concept in the book, and that is the book talks a lot about Mendel's peas. So this is this monk, Gregor Mendel, from the late 1800s, did a lot of experiments on peas. And he looked at 7 traits, 7 phenotypes, and it happened that they were on the 7 different chromosomes of peas. So that those traits would assort, they would separate independently of each other. And you could send on one of the other characteristics. So a couple of things. One is, the book we'll see is, what if it happens that 2 genes are on the same chromosome? So we're going to learn a little bit about that. If they are physically linked, and that's the term also, that they're physically linked. So when they are go through meiosis, they don't separate, they stay together. So what if they're linked? And then what if we look at, just in general, what if we look at 2 traits? So we have been looking at what's called a monohybrid cross, just one gene. And what if we looked at a dihybrid cross? And the answer is that the traits would either be on separate chromosomes or the same chromosomes, and we'd have a different Punnett square depending on those 2 possibilities. So we're going to have Punnett squares, and they're just going to be a little bit more complicated with a little bigger ratio than the monohybrid 3:1 and 1:2:1 ratios. But we're going to come up with some theoretical ratios based on the law of independent assortment and segregation, and look at those numbers as well. So we just could take 2 traits, seed color, seed shape, If they're on separate chromosomes, they'll separate independently, and we'll have this kind of Punnett square. And we come up with a theoretical ratio of this. And then here's a practical result in like a field experiment, a breeding experiment out in a big garden with peas. And it's just bringing up this idea that we learned about one gene. What about two genes? And in this case, the genes are on separate chromosomes. And that's just shown here. So I tried to draw this out here to give you a visual, add this to the book, that we're looking at the law of independent assortment and segregation based on the two traits that are on different chromosomes. Okay, now what if they're on the same chromosome? The answer is the Punnett square gets simplified because these traits are linked and these traits are linked, and they will excuse me, segregate together. So we can't get all the combinations. They'll segregate together, and we have a simpler phenotypic ratio. The ratio looks very much like a monohybrid cross because we are really sort of making just two comparisons here. And you want to work through this, look through this idea, but it's a pretty straightforward idea, and you just want to be solid on it. Then, described back again in this chapter on meiosis, we learned about the idea of recombination very briefly. At that point in our course, in our lecture, we just learned that that occurs in meiosis 1. Recombination occurs. Meiosis 1 is a crossing over randomly between dads and moms homologous chromosomes, so that at the end of meiosis 1, we have some chromosomes that look like this. And this recombination can affect linked genes. So we started out, if they're unlinked, we saw a complex Punnett square with the ratios of 9 to 3 to 3 to 1. If they're linked, that ratio simplifies 3 to 1. And so we see that in the field. Now, sometimes in a field, we'll see, this thing where it's basically 750 plants that look like they have yellow, smooth seeds like that, and 250 plants about that have green, wrinkled seeds, that those traits are linked, but that there'll be a low frequency. This recombination is a low frequency, like maybe 1 in 1,000 or 1 in 500. So there might be like 5 plants, and this would be dihybrid. Only in dihybrid you would see this, where you would have like yellow, wrinkled, and you'd be like, hey, what's going on there? So it's about that frequently, and it is the result of meiotic recombination. So we come up with these ratios, and there could be a rare event like this leading to, and it would be a dihybrid. So dihybrid cross, and these are not new alleles, not new mutations, right? At a lower frequency, maybe one plant, you might see a plant that is like, what would it be? Like, yellow, wrinkled, green, smooth. You might see a plant that would be sort of like, let's just say like dark green, wrinkled. And so it's that same trait, seed color, but it's a new color and it's rare, and that could be the rise of a new mutation, very rare. So in a field, we look at a field of plants in this cross, and the first thing we look at is there are going to be these large general phenotypic ratios that are going to help us determine the relationship between these alleles: codominant, incomplete dominance, dominant recessive. Then meiotic recombination in dihybrid crosses, where we're watching two alleles that are linked, can yield some recombined phenotypes, where we could be like, wow, we don't see green smooth very often, but here we are seeing it at a low frequency, probably due to linkage. And that linkage allowed scientists to map positions of genes along the chromosomes, and so this linkage and recombination was very helpful historically in genetics. So it's in our book because it was helpful historically, especially, and well, because it's true biologically, and also it's quite helpful to genetics historically. Good. So then, just last thing is to pick up these stories from meiosis 1 and meiosis 2 in chapter 13. So it's an image from chapter 13, and we've got these 3 important events that happened during meiosis 1 and 2. This is a review from chapter 13. Boom. That recombination, this crossing over of linked genes, occurs during metaphase of meiosis 1 by chance, randomly, and relatively rarely. Then sister chromatids, these identical photocopies, stay together during anaphase of meiosis 1. There's no S phase between meiosis 1 and meiosis 2, so we go from tetraploid all the way down to haploid, and this would be, sperm or eggs. In males or females. That's that. Okay, good. And so we are all set with these pieces.